Okay, this is a lecture on the integumentary system. So if we look at the functions of the integumentary system, we see the vital functions include protection from radiation, that's ultraviolet radiation. You remember melanin does that. Protection from chemicals, toxic chemicals are not easily absorbed through the skin. Protection from pathogens, and I think most importantly, protection from fluid loss, because if you lose that waterproofing barrier of the epidermis, then you weep tissue fluid, and it can, if you weep too much out, it can kill you. You also have temperature regulation through sweating and flushing, and of course sensory awareness of when you're too hot or too cold, and that leads to sensation. We also have pain, sensation, and tactile sensations, and you're assigned to learn the sensory nerve endings. You also have the non-vital functions of vitamin D production. Excretion of waste is usually listed as a skin function, but I think it's kind of incidental. It's like, it's just there, it's not really a specific function. Looking at the histology of the skin, look on the right side of the diagram, and here you see our two basic layers, the epidermis and the dermis. There's also a hypodermis. The hypodermis is a loose connective tissue, usually full of adipose, depending on how much adipose you're carrying around. And the hypodermis is not technically considered part of the skin. It's a separate layer underneath, and it's also called the superficial fascia, or the subcutaneous layer. So if you put a hypodermic needle into the subcutaneous layer to give a sub-Q injection, you're going under the skin. So we know our basic layers are the epidermis and the dermis. The epidermis you learned is a keratinized stratified squamous epithelium. It's got 4 distinct types of cells and has 4 or 5 distinct layers or strata. The dermis has 2 layers. The papillary layer is an areolar connective tissue. Remember, that's a loose connective tissue. And the reticular layer, the deeper part, is irregular, dense connective tissue. It's a mix of collagen fibers, mostly collagen for puncture resistance, and then some elastic fibers mixed in. Additionally, in the skin, we have the accessory structures that includes glands, hairs, and then, of course, blood vessels and nerve endings, and then the hypodermis, which, of course, is not part of the skin. In this diagram, actually in these 2 diagrams, you can compare the thick skin with the thin skin. They've made them relatively the same thickness, and that's kind of unfortunate. But what you see in thin skin that you don't see in thick skin is that there are hairs. There is hairless skin besides your thick skin, the distal phalanges, your lips, and some other areas that don't have hairs. But you can also see in this diagram that you have a great blood supply. Take a look at the diagram on the left. And here you can see in the deepest part of the skin, the blood vessels that arrive to supply the entire skin. This is called here it's called the deep arteriovenous plexus. I usually call it the cutaneous plexus, because that's the main supply to the skin. And then look at what happens as those blood vessels from that cutaneous plexus go up through the dermis, and they have this nice rich capillary network up here in the papillary region of the dermis, and that up there, what this book calls the superficial, we call the papillary plexus. So you have a deep cutaneous plexus and you have a superficial papillary plexus. You've also got a variety of nerve endings. You've got hair follicles and the muscle to supply the hair, and you've got the glands. We'll come back and talk about each of those in turn. Okay, so we're going to focus first on the epidermis. In this nice micrograph, you can see quite clearly there are layers to the epidermis. So you see that very darkly pigmented stripe running through, about halfway through, and that distinguishes the living tissues below from the dead tissues above, or dead layers above. This is the only kind of epithelium in which we name the cells. The cells of the epidermis are called the keratinocytes, because this is the only place we find a keratinized stratified squamous epithelium. The purpose of keratin is to help make the epidermis more water resistant, and keratin is a mix of protein and lipid. It's very hydrophobic. The other main cell type in the epidermis is the melanocytes. They produce the pigment melanin, which absorbs ultraviolet radiation. We'll talk more about that next. So as you look at this figure, you see those two main cell types, the melanocytes, and everything else that's kind of pink is the keratinocytes. So let's focus on the melanocytes first. Melanocytes produce the melanin, and they produce it in little packages called melanosomes. So here you see the little melanosome packages. The melanin can escape from the melanosomes, and so you can see the little granules of melanin here. And for the most part, unlike the way it's shown in this picture, the melanosomes are going to sit above the nucleus of the cell, and that's going to help protect it from UV, because the UV radiation is coming through from the outside. So think of the melanosome as being like a little protective cap, a hat, or a parasol that protects the nucleus from ultraviolet radiation damage, because UV damage can cause DNA mutations, which could cause the cells to become cancerous. So in darkly pigmented skin, you see there's just way more pigment than there is in lightly pigmented skin. The melanosomes are being pinched off of these long cellular processes that just run between the living keratinocytes. Most of the cells in the picture, of course, are the keratinocytes. They are, unlike the way the picture depicts them, they are connected with lots of desmosomes. All of these cells are interconnected with desmosomes. As the cells age and die, as they move up, the desmosomes tend to break down and the layers are going to flake off. Okay, there are two other cell types found in the epidermis. The Merkel cells are more commonly called tactile cells. We'll see a picture of that in just a sec. The Langerhans cells, I don't have a picture of. These are called dendritic cells, and they're responsible for your immune reactions to anything that gets into the living part of the epidermis. They're not going to react to anything in the dead layers. So if you look at what's going on in the Langerhans cells, they're just going to kind of squeeze in between these cells, and they're going to wander around just in the living part down here. So if a chemical or a microbe managed to sneak its way through, that Langerhans cells would react to it, and its reaction is going to be to signal changes in the dermis that's going to cause inflammation, which is going to be redness and potentially swelling. All right, so that's it on those two cell types. We'll look at the Merkel cells more later. So here are the epidermal strata from top to bottom. The corneum at the top, that's the outer layer of dead cells. The lucidum, which is found only in thick skin. The granulosum, which is that layer that is darkly pigmented. And then below that, the living stratum spinosum and stratum basale. So as we look at those layers, in this diagram, you can see the stratum basale is just that single row of cells that connects by hemidesmosomes to the basement membrane. Above it, you've got a layer here, the stratum spinosum, which most books will say is 2 to 8 cell layers thick. That looks like more than 8 to me. This darkly pigmented layer, the stratum granulosum. Above it, that very clear, thin pink layer, the stratum lucidum, again found only in thick skin, and then above it the thickest layer, which is the stratum corneum, and all of those cells are little dead bags of keratin. So here we see it in a diagram, and you can see that the cells at the bottom, the stratum basale, they're attached to the basement membrane by hemidesmosomes. Here you see little teeny tiny lines indicating the desmosomes. Here's your melanocyte with its cellular processes delivering melanin to all of the cells around it. Here is that Merkel cell, that's the tactile cell. It just kind of plugs into a nerve ending here, and so it responds to very light touch because it's as superficial as you can get. The cells of the stratum spinosum can go through cell division. The stratum basale definitely, that's where most of the cell division occurs because they generate the layers above. But you can still have some cell division in the stratum spinosum. And then when you get to the stratum granulosum, it's called granulosum because these cells have these little granules in them. So you see the little tiny spots in each one. And what happens is as those cells reach the top, they dump all their granules. So a cell here that used to be in this layer has dumped all its granules, and those granules are dumped underneath of itself. So you cannot see it in the picture, they don't really show it, but if you look at the cells above that one, well, that cell sits about here, and it had already dumped its granules. So you get these layers of cell, keratin, cell, keratin, and eventually those cells, well, they're dead as soon as they dump their granules, and the reason for that is you've got a layer of keratin here. You have another layer of keratin here, of course, and so forth all the way up. Keratin, remember, waterproofs. Well, where's the blood supply for these cells? The blood supply comes from the dermis. So if my blood vessel is here, then diffusion of nutrients is going to supply mostly the stratum basale and the lowest layers of the stratum spinosum, and the nutrient diffusion gradient is going to die out as the living cells of these deeper layers are going to pick up most of the nutrients. So by the time you get out to the stratum granulosum, hey, you're not very close to the blood supply, and these cells are busy making keratin. So once they've dumped their keratin underneath of themselves and move into the next layer, they're dead. All right, so to summarize the characteristics of each layer, the stratum corneum is that superficial most layer. It's the thickest, and it's dead. Those cells are dead, dead, dead. The stratum lucidum, which is found only in the thick skin of the palmar and plantar surfaces, is that clear layer. Again, those cells are dead, but I don't even know why it's there, but it's clearly distinct on histological preparations. Stratum granulosum, those are the ones that stain darkly. That's the layer in which the cells are going to die. Deep to that, the stratum spinosum is the thickest layer of living cells. Some cell division can occur here, and because the cells can divide, if a mutation occurs, if some of that ultraviolet radiation gets through or some toxic chemical gets absorbed and mutates the DNA, those cells can become cancerous. If they're in the stratum spinosum, remember they're not attached to the basement membrane. These cells can migrate. That's a squamous cell carcinoma. In the stratum basale, that's your deepest layer. These are the ones that are attached to the basement membrane, so even if they do become cancerous, and that would be a basal cell carcinoma, they tend not to migrate so much. Okay, moving on to the dermis. In the dermis, we have our two layers. We have the upper thin papillary layer, and we have the deeper thick reticular layer. So looking at it on this diagram, the dermal papilla, that's a papilla there. So this layer just at the very top is the papillary layer. It's a loose connective tissue. It's got the blood supply for the epidermis. It has some nerve endings. And remember that it's folded to increase the surface area. And again, that increased surface area is better for attachment between the epidermis and the dermis, and it also increases the surface area for diffusion of nutrients up to the epidermis. The thicker, deeper layer of the dermis, the bottom probably 80%, is the reticular layer. That is our irregular, mostly collagenous, dense connective tissue. All right, so here's a summary of those layers. The papillary layer, because it's areolar, it's a very fragile tissue, it's easily torn, so you're not going to make a thick layer there, but that does have the blood vessels. It's well vascularized to supply the epidermis, and it also has some specialized nerve endings, which we'll meet later. The reticular layer, because it's mostly collagen, is pretty puncture resistant, but because it's the main thickness of the skin, that's where most of the accessory structures are, like hairs and glands. The fibers of the irregular collagenous tissue are kind of arranged every which way, but there's always a predominant direction. So look at the palmar surface of your hand. Look not at your hand, but your fingers. Look at the longitudinal kind of lines that run along the ventral surface of your fingers. Those are lines of cleavage. And the lines of cleavage are simply the predominant direction of collagen fibers in that part of the body. And we map these because surgeons need to know which way to make incisions. So as long as an incision runs along the direction of the lines of cleavage, then when you cut it, it tends to close up pretty well. If you go across the lines of cleavage, then that wound tends to gap open. So if you've ever cut your finger across with a knife, then you know you get an uglier scar when you cut across the cleavage lines rather than along the cleavage lines. Okay, so now we're going to focus on our specialized structures. This includes the hairs, but more importantly the glands. And we're going to start with the dermal papillae. We do consider them structures of the dermis. Again, they're highly folded. That gives them a lot of surface area. It's that thin, fragile, areolar connective tissue that contains the capillary network to supply the epidermis. And again, that capillary plexus is the name of that. And we also find some specialized nerve endings for tactile sensation. These are called tactile corpuscles. Don't get them confused with the tactile discs, which we've already met, or the tactile cells. So as you study the nerve endings, you got to keep the names straight. Okay, so here is an image from a woman named Sherry Amzell. There's her web page there where I borrowed this slide. And you can kind of get a sense of what the different kinds of nerve receptors are. Thermoreceptors for sensing temperature. Here are those specialized receptors, the tactile corpuscles that are found up in the dermal papillae. You've got nociceptors to sense pain. And then you have these other specialized guys, the Pacinian corpuscles. Now, these all have other names. Pacinian corpuscles, though, are very deep, so you kind of have to pinch or poke or prod someone pretty hard to get them activated. On the other hand, they also get activated when you get shaken with vibrations. So if you're ever in an earthquake, your Pacinian corpuscles will let you know. Missing from this picture is the first little guy we met, which was just a little tactile disc, tactile cell up in the epidermis. And the nociceptors are just free nerve endings. They're not specialized, and they can go up in the epidermis as well. So you can feel pain, say from a paper cut that's so shallow it doesn't make you bleed because you've just cut into the epidermis, but it still hurts, so you know you got pain nerve endings in your epidermis. All right, accessory structures found in the deeper dermis include the hair, the nails, and some glands, and the main glands are the sweat glands and the oil glands. Hair is considered an organ. Actually, the hair itself is a non-living structure. It's the hair follicle that's the living organ. And the hair follicle does contain some stem cells from which new epidermis can arise. So if you have a patient who's been burned deep enough to burn off all the epidermis and maybe the upper parts of the dermis, the epidermis can grow back from stem cells in the hair follicles. There's also, of course, a blood supply and a nerve supply to the hair follicle, and the sensory receptors are just called hair follicle receptors. Those are the ones that let you know when your hairs get moved, either by the wind or because a mosquito lands on your arm. Why did we do that? Okay, and then the smooth muscle that moves the hair is called the erector pili muscle. It's going to make your hair stand up when you're cold or stressed or scared or angry, and that's all part of your fight or flight response. Don't test you on the structure of nails. I do want you to be aware of nails as a diagnostic tool. Not only can you tell some disorders, for example, in the right you see the club nails, those are associated typically with heart disease, although we don't really know why. But even the color of the nail bed can be diagnostic. So if you press on one of your nails, the nail bed turns white because you're squeezing all the blood out, and then if it revascularizes, it turns pink. So if you look at the nails of a patient, then you get a good sense of their circulation. There are two major kinds of glands, the sweat glands and the oil glands. There are two kinds of sweat glands, though, merocrine and apocrine. Merocrine glands are often called eccrine glands, and it's the merocrine glands that are doing the sweating associated with cooling you off, the thermoregulation. Sweat is also slightly acidic, not real acidic, but that does inhibit bacterial and fungal growth on the surface of the skin. So you have merocrine glands distributed throughout the body, throughout the skin. So you have millions of those. The apocrine glands are specialized glands. They're found in the axillary and inguinal regions, and we associate them with the production of pheromones. So if you've never heard of pheromones, a pheromone is a substance, a chemical secreted by one individual that can be sensed and responded to by another individual. So for example, every male female dog in the neighborhood knows when a certain female dog goes into heat, or every male moth in a cornfield can find the female moth when she's ready to mate. Those are pheromones. Human pheromones are suspected but not proved, but there's evidence that human behavior, particularly sexual behavior, can be triggered by the presence of pheromones. Oil glands, or more properly called sebaceous glands, they lubricate the skin, and that prevents the skin from cracking. This is important if you go into healthcare and you have to wash your hands a lot. You want to keep your skin lubricated because a crack in the skin can be an entry point for an infectious organism. And again, that acidic pH inhibits bacterial and fungal growth. Okay, so in this diagram you can see the glands. The main kind of sweat gland here, the eccrine or merocrine sweat gland, has a duct. It opens up onto the surface of the skin. Sebaceous glands are typically associated with hair follicles, but on hairless skin they can open directly to the surface of the skin as well. There are two other kinds of glands that are specialized. They're actually specialized from the sweat glands. The ceruminous glands are the ones you find in the ear canal. They secrete earwax, and yeah, it really does repel insects. It's kind of bitter and sticky, so it keeps bugs out of your ears. The mammary glands are the glandular tissue within the breast. So the mammary glands themselves are not the entire breast, they're the glandular tissue within the breast. And they don't mature unless a woman becomes pregnant. So if a woman dies in her 90s and never had kids, she's never going to have active mammary gland tissue. And obviously these are the glands that produce milk, and I suppose that makes milk modified sweat, although that doesn't sound very appetizing. Okay, so we're going to move into some of the physiology of the skin, and as we move into this section we'll be combining a lot of the structures. So with thermoregulation, anything that threatens homeostasis, whether you're too hot or too cold, can trigger a fight or flight response. That activates an autonomic response. And we'll learn more about that when we get to the autonomic nervous system, or to the nervous system. But you probably have an intuitive sense of this, since you can be consciously aware of sweating, and you can be consciously aware of when your blood vessels dilate or constrict. So you have thermoreceptors in the skin that send signals to, there's a part of the brain that's responsible for regulating your temperature, and if you're too hot, it's going to activate both those merocrine sweat glands and to dilate the cutaneous blood vessels, and you get evaporative cooling as sweat evaporates. And you get what we call radiant cooling, when heat just radiates out through the blood vessels. That heat radiates right through your epidermis out to the air. That doesn't work very well if the air temperature is higher than your body temperature. This can cause problems for people with maintaining body temperature, and we see this particularly in heat waves. So if you live in Phoenix, even though it's a dry heat, you get a lot of evaporative cooling. If the air temperature is too high, it's really hard for radiant cooling to do much good. Obviously, you're going to do the opposite of that if you're in cold. When I ask students what the skin's response to being too cold is, usually the first words out of their mouth are, you shiver. Well, yes, you do shiver, but that's a muscular response. It's not a skin response. So the skin response is to, well, first of all, not to sweat, but more importantly, to constrict those cutaneous blood vessels, and that's going to reduce the heat loss through the skin. Now, you can't do that forever, because your skin does need to have a blood supply. So what happens eventually, if you're in intense cold, is that the skin dies, and this is frostbite. All those blood vessels are going to shut down until you get back into some warmth, if they can. So here is a diagram again showing you that papillary plexus in the upper layer, cutaneous plexus in the lower layer. It's the cutaneous plexus that's going to control thermoregulation. It's going to cut off blood flow in the cold or open up those blood vessels in the heat. When people get old, they don't thermoregulate very well. First of all, a lot of them can't tell if they're too hot or too cold. Second of all, they're not very good at retaining heat or losing heat. And so you're more likely to lose elderly individuals in extreme weather events, whether it's from they got too hot, or there was a cold snap and they didn't have heat in their apartment, hypothermia may set in. So elderly individuals, basically as you age, things slow down, and so everything works more slowly in the elderly. Okay, so that was the function of thermoregulation. In vitamin D production, we talked about this in class already. We know that vitamin D is produced from cholesterol, so it's a steroid. We know that it requires ultraviolet radiation for one of the metabolic steps, and that means it has to happen in the skin. So that's the only place in the body where UV radiation is reliably going to come. So you do the first chemical step of turning cholesterol into vitamin D in the skin, and then it gets into the circulation. It goes to the liver. It's processed a little more. It goes from the liver to the kidneys, and synthesis of the final version, the final product, is a hormone called calcitriol, and calcitriol is important for calcium homeostasis. So we'll talk about that as we get into the next chapter, although I know I already did it in class a little bit. So calcitriol, because it's a steroid, prompts cells to produce a protein. That's what steroid hormones do. So a deficiency of vitamin D means you don't have enough calcium absorption from diet, and you can't keep it from being lost in the urine from the kidney cells. They're just not good at reabsorbing the calcium, so you wind up taking more calcium out of your bones. And this calcium deficiency in the bones usually, again, occurs in the elderly. They're on a fixed diet, they don't get enough sunshine, and so they get a condition which I remember writing down in class called osteomalacia. So this is what we see in the elderly typically if they're on a limited diet and they're not getting enough vitamin D. And in children, it leads to rickets. Those are your bone calcium deficiency diseases of bone. And we keep finding more things that vitamin D does for us, so stay tuned to the vitamin D story. Okay, so we're going to talk a little bit about normal and pathological skin color. What you're seeing in this individual is a condition called vitiligo. Vitiligo is an autoimmune disease in which the immune system inappropriately attacks the melanocytes, those pigment-producing cells. It's more exaggerated in people with darker pigmented skin, but it's also more common in people with darker pigmented skin. So natives of Africa or North American natives, indigenous North Americans. Okay, so we have some normal skin pigments, and the main one, of course, is melanin. But we also see carotene being absorbed in the skin. Carotene is a yellow pigment. Yes, it's what makes carrots orange and corn yellow, and it's a hydrophobic little chemical, and we don't make it, but if we eat a lot of it, then it can get absorbed and parked in the keratin. Don't get carotene and keratin confused, but they hang out together. So if you've ever had a callus on your finger and you notice it's a little bit yellow looking, that's why. It's not that common, but sometimes little kids, toddlers that are being introduced to new foods, if they decide they only want to eat sweet potatoes and nothing else, which is not that uncommon because they're sweet, they can actually turn yellow from eating all that sweet potato, all the carotene in it. And then the third normal skin pigment is hemoglobin, and we, of course, don't leave it in the skin. It's just in the circulating blood, but it's important for diagnosing certain cardiovascular disorders. And so here are the abnormal colors associated with cardiovascular disease. Erythema is reddening. It can be from flushing of the skin. It can be from fever. It can be from inflammation. It could just be from embarrassment. If you have spots like a rash, that's still erythema, but we have specific terms for that. We're not going to get bogged down in those in this class. If the skin looks blue, that's called cyanosis. And that's from the blood circulating, it's just not well oxygenated. So oxygen attached to hemoglobin makes it just shine brighter, so it's a bright red. And if it doesn't shine very well, then it just doesn't shine through the epidermis or the dermis very well. And that makes the skin look blue or cyanotic. Pallor is pale, and that's lack of blood flow, typically from hemorrhage. I have no idea what's causing these two individual cases that I've snagged these pictures from, because that's kind of scary. If you think that typically you're just going to turn pale from hemorrhage, that the individual with those fingers has obviously a local problem going on, and that could be just a response to low blood flow. All right, now there's some abnormal pigments, things that don't usually show up in the skin, and the primary one you need to know is bilirubin. Bilirubin is a breakdown product of red blood cells, hemoglobin. So as hemoglobin is processed at the end of a life of a red blood cell, the pigment part, the part that's red in the red blood cell, gets converted to this other pigment called bilirubin, which when it's concentrated looks really green. That's what makes bile green. But when it's more diluted out, like when you see it in the skin of an individual, it looks yellow, and that's what jaundice is. It's a yellowing of the skin. If you see it in an adult, it's typically a sign of liver disease. If you see it in a newborn, it's temporary. It's because their liver's not mature yet, but that excess of bilirubin can damage the brain. So we always treat newborn jaundice quickly. It's treated very quickly. Cool thing is bilirubin gets broken down by ultraviolet light. So if you have a jaundiced newborn, you can take them outside and just get them some sunshine, but they actually produce these artificial lights called Billy lights, specifically to treat the newborns with jaundice. It's not something to ignore. Okay, so when you get finished with this chapter, review all the functions of the skin. Make sure you can associate each part of the skin with the function that it carries out. Go through the chapter summary, go through the integration lesson, and specifically on the tissues part of the integration lesson, and do all of that before your next test. I believe that's the end of our integumentary system review.